institutions already involved, both in Croatia, in UK, even China. We have Wei here from China, who's also part of it. So I think we are making at least some steps forward. So what we are trying to understand in the moment is genetics of protein glycosylation. Because contrary to protein synthesis, where you have a single gene, which will give you maybe 1, maybe 10 proteins with different alternative splicing models. Now you have 500 genes working together to give you one glycoprotein. But not only this one, but also all the other glycoproteins will be produced by a number of genes which work together. And we know some of them. We know glycosyl transferases, we know glycosidases, but we don't know the others. We don't know the transcription factors, we don't know the ion channels, we don't know the Golgi organisms, all other proteins which are involved. Very indicative study was done nearly 2 years ago in Japan. They were doing a large RNAi library inhibition of neuron-specific glycosylation in Drosophila to see which genes are needed to produce normal glycosylation in Drosophila, in neuron-specific glycosylation in Drosophila. They identified 109 genes. Only 14 of these genes were the known glycogenes. All other genes were some other genes. So you cannot directly extrapolate because many of those other genes in signal transduction, protein degradation, they will be important for all other types of glycosylation. But I think we can be pretty safe in estimating there's something like 2,000 genes working together to glycosylate proteins. So we're talking about 10% of the genome in single metabolic pathway, which is by far the most complex metabolic pathway we have. And we know very little about polymorphisms in those genes. And this is actually not surprising, because about common polymorphisms in any disease, we started to learn only a few years ago when GWAS study started. What we know for the glycogenes, we know the CDGs, which either you're dead or you have a leaky mutation. We know the ABO blood groups, but we don't know much about it. But there was this revolution in genetics a few years ago when the GWAS studies were introduced. So people started to look into large population cohorts, look at all genes, and ask the question not, is this gene important? The question was, which genes are important? And this apparently worked. Because when you look into mostly cited papers in Nature, and this is Igor's slide, a few years ago, all those marked with the red arrows are the GWAS papers. So this is something which is being published, which is being published in top-level journals, which is being cited a lot. And fortunately, at the same time, Pauline developed the high-throughput method for glycan analysis. So it was possible to join glycomics and genomics. And we were close enough to jump on the first ship. So we did the first large study of the glycome in humans. It was done in a collaboration between Pauline's and my group. And we published it a few years ago where we showed there's a large variability in human population. And we were actually doing just relatively simple analysis. You cannot analyze all thousands and thousands of different glycan structures. But we did several HPLC runs. And HPLC is good in grouping glycans in similar structures. So we could deduce a lot of information from those few HPLC runs per person. So what we learned, that the glycans are extremely variable between different individuals. But then, in the same individual, they're very stable. So they're more or less predefined. There are slight changes with time, but individual glycome is very stable. Between individuals, there are lots of differences. And there are some individuals which are very different. We call them outliers because they have, this is the normal profile, this person has a huge, this biantenary monocell structure. This person here had no neutral glycans, or you have different other aberrations. And there are groups of people which have some different glycotypes, which is obviously some kind of a genetic or environmental reason. So, as I said, we can't do it for all possible glycoforms. Even for IgG, this would be very complex. But you can do it in a quantitative way, get some glycotraits, which are specific for individual, and then you can try to put this thing in the GWAS studies. And this is our first attempt. And I think we had only 1,000 people, and we got a very nice peak in here. this was not a fascinating discovery because this was a FUT8 gene, and we found it's regulating fucosylation of this one of the glycans, so it was logical. But this was a proof of principle that it works. So actually, we didn't even publish it. We posted it in Nature Proceedings as a proof of principle that single SNP can have a significant effect on a single glycan. So these things work. We can use glycomics to go to GWAS studies. And then when we analyzed more people, we did the first large GWAS study of the glycome, and we found the first interesting hit, which is HNF1 alpha. And it was linked to a number of glycan structures, mostly related to the absence or presence of antennae fucose. And then with a lot of functional genomics work done in MRC in Edinburgh, it was shown that this gene is actually controlling the number of glycosyl transferases, and that actually this is the master regulator of antennae fucosylation in human blood.